Ammonium fertilized early life on Earth. A team of international scientists, including researchers at the University of St Andrews, Syracuse University and Royal Holloway, University of London, has demonstrated a new source of food for early life on the planet. Life on Earth relies on the availability of critical elements such as nitrogen and phosphorus. These nutrient elements are ubiquitous to all life, as they are required for the formation of DNA, the blueprints of life, and proteins, the machinery. They are originally sourced from rocks and the atmosphere, so their availability to life has fluctuated alongside significant changes in the chemistry of Earth's surface environments over geologic time. The research, published in Nature Geoscience, reveals how the supply of these elements directly impacted the growth of Earth's oxygen-rich atmosphere and were key to the evolution of early life on Earth. The most dramatic change in Earth history followed the evolution of oxygenic photosynthesis, which fundamentally transformed the planet by providing a source of carbon to the biosphere and a source of oxygen to the atmosphere, the latter culminating in the Great Oxidation Event, GO, some 2.3 billion years ago. Despite the critical importance of nutrients to life, the availability of nitrogen and phosphorus in pre-GO oceans is not well understood, particularly how the supply of these elements drove and/or responded to planetary oxygenation. Using samples of exceptionally well-preserved rocks that have been associated with early evidence for oxygenic photosynthesis 2.7 billion years ago, the team of researchers examined Earth's early nitrogen cycle to decipher feedbacks associated with the initial stages of planetary oxygenation. There is precious little rock available from this time interval that is suitable for the type of analyses we performed. Most rocks that are this old have been deformed and heated during 2.7 billion years of plate tectonic activity, rendering the original signals of life lost, says Christopher Junium, Associate Professor of Earth Sciences in the College of Arts and Sciences. The rock samples showed the first direct evidence of the buildup of a large pool of ammonium in the pre-GO oceans. This ammonium would have provided an ample source of nitrogen to fuel the early biosphere and associated oxygen production. Research team leader Aubrey Zirkle, reader in the School of Earth and Environmental Sciences at the University of St Andrews, says, "Today we think of ammonium as the unpleasant odor in our cleaning supplies, but it would have served as an all-you-can-eat buffet for the first oxygen-generating organisms, a significant improvement on the dumpster scraps they relied on earlier in Earth's history." As well as helping scientists better understand the role of the nitrogen cycle in global oxygenation, the new findings also provide context for other nutrient feedbacks during early planetary evolution. It is becoming ever more clear that the game of nutrient limitation has tipped back and forth through Earth's history as life has evolved and as conditions have changed, Junium says. Surprisingly, evidence for significant atmospheric oxygenation does not appear until 400 million years later, meaning that some other nutrient, such as phosphorus, must have been important in setting the evolutionary pace. Eastern forests shaped more by Native Americans burning than climate change. Native Americans' use of fire to manage vegetation in what is now the eastern United States was more profound than previously believed, according to a Penn State researcher who determined that forest composition change in the region was caused more by land use than climate change. "I believe Native Americans were excellent vegetation managers and we can learn a lot from them about how to best manage forests of the U.S., said Mark Abrams, professor of forest ecology and physiology in the College of Agricultural Sciences. Native Americans knew that to regenerate plant species that they wanted for food, and to feed game animals they relied on, they needed to burn the forest understory regularly. Over the last 2,000 years at least, according to Abrams, who for 3 decades has been studying past and present qualities of eastern U.S. forests, frequent and widespread human-caused fire resulted in the predominance of fire-adapted tree species. And in the time since burning has been curtailed, forests are changing, with species such as oak, hickory and pine losing ground. The debate about whether forest composition has been largely determined by land use or climate continues, but a new study strongly suggests anthropogenic fire has been the major driver of forest change in the East, said Abrams. That is important to know because climate change is taking on an ever larger proportion of scientific endeavor. But this phenomenon does not apply to other regions, Abrams noted. In the western U.S., for example, climate change has been much more pronounced than in the East. That region has received much more warming and much more drought, he explained. Here in the east, we have had a slight increase in precipitation that has ameliorated the warming, said Abrams. To learn the drivers of forest change, researchers used a novel approach, analyzing both pollen and charcoal fossil records along with tree census studies to compare historic and modern tree composition in the forests of eastern North America. They looked at 7 forest types in the north and central regions of the eastern United States. Those forest types encompass 2 distinct floristic zones, conifer northern hardwood and sub-boreal to the north, and oak pine to the south. The researchers found that in the northernmost forests, present-day pollen and tree survey data revealed significant declines in beech, pine, hemlock and larches, and increases in maple, poplar, ash, oak and fir. In forests to the south, both witness tree and pollen records pointed to historic oak and pine domination, with declines in oak and chestnut and increases in maple and birch, based on present-day data. Modern forests are dominated by tree species that are increasingly cool-adapted, shade-tolerant, drought-intolerant pyrophobes, trees that are reduced when exposed to repeated forest burning, Abrams said. Species such as oak are largely promoted by low to moderate-level forest fires. Furthermore, this change in forest composition is making eastern forests more vulnerable to future fire and drought. Researchers also included human population data for the region, going back 2,000 years, to bolster their findings, which recently were published in the Annals of Forest Science. After hundreds of years of fairly stable levels of fire caused by relatively low numbers of Native Americans in the region, they report, the most significant escalation in burning followed the dramatic increase in human population associated with European settlement prior to the early 20th century. Moreover, it appears that low numbers of Native Americans were capable of burning large areas of the eastern U.S. and did so repeatedly. After 1940, they found, fire suppression was an ecologically transformative event in all forests. Our analysis identifies multiple instances in which fire and vegetation changes were likely driven by shifts in human population and land use beyond those expected from climate alone. Abrams said. After Smokey Bear came on the scene, fire was mostly shut down throughout the U.S. and we have been paying a big price for that in terms of forest change. We went from a moderate amount of fire to too much fire to near zero fire, and we need to get back to that middle ground in terms of our vegetation management. Also involved in the research was Gregory J. Nowacki, with the Eastern Regional Office, U.S. Department of Agriculture Forest Service. The Agricultural Experiment Station of Penn State funded this research. Scientists propose rethinking endangered species definition to save slow-breeding giants. Conservation decisions based on population counts may fail to protect large, slow-breeding animals from irrevocable decline, according to new research coinciding with Endangered Species Day. Critical thresholds in so-called vital rates, such as mortality and fertility rates among males and females of various ages, can signal an approaching population collapse long before numbers drop below a point of no return, says lead author Dr. Sherman De Silva, president and founder of Asian elephant conservation charity Trunks and Leaves. We propose that conservation efforts for Asian elephants and other slow-breeding megafauna be aimed at maintaining their demographic safe space, that is, the combination of key vital rates that supports a non-negative growth rate. A Mammoth Insight. Published in Frontiers in Ecology and Evolution, the study suggests that a combination of key vital rates governing population growth is a better indicator of a species' viability than short-term trends in population size and distribution. History bears this out, argues De Silva. Genomic studies of the last mammoths isolated on Wrangel Island, between Russia and Alaska, have shown that although they were able to persist for thousands of years beyond the extinction of mainland populations with just approximately 300 individuals, they had accumulated numerous genetic mutations that may have eventually contributed to their extinction. In other words, populations of megafauna can become biologically inviable long before they disappear, if pushed beyond their demographic safe space. Females and calves key to saving the Asian elephant. The group applied the demographic safe space concept to the case of the Asian elephant. Asian elephants are classified as endangered under the IUCN Red List because populations are thought to have declined by at least 50% in less than a century, explains De Silva. There are fewer than 50,000 wild Asian elephants living today. Studies show that wild Asian elephants breed extremely slowly, the majority producing just one calf in 6 years or more. Using mathematical modeling, Da Silva and colleagues found that near-optimal reproduction and high calf survival is necessary to maintain non-negative population growth in the face of even modestly increased mortality among adult female age classes. The approach shows a clear conservation priority for Asian elephants, a species in which the vast majority is tuskless. Measures to enhance survival of calves, and particularly females, are key to saving the Asian elephant, emphasizes Da Silva. But while the attention of the world has been focused on the ivory trade, for critically endangered Asian elephant populations the greatest threat is habitat loss, followed by illegal trade in live animals and parts. Habitat loss can create something known as extinction debt by slowing down birth rates and increasing mortality rates. For slow-breeding long-lived species, even incremental changes make a big difference, but their longevity can obscure the risk of extinction. A demographic safe space for all megafauna. Conservation efforts for other large, slow-breeding species, such as giraffes, rhinos, Bactrian camels and eastern gorillas, could also benefit from modeling the interaction between vital rates. Data for these species in the wild are a scarce yet urgent necessity, suggest the authors. Rather than rely on simple population counts or estimates of near-term extinction probability, we urge that conservation resources for slow-breeding megafauna also be invested in identifying demographic tipping points and how to maintain populations within their safe spaces. Populations of slow-breeding taxa need proactive management well before numbers become critically low, when returns on investment are potentially greater and populations less likely committed to extinction, concludes Da Silva. Bedbugs evolved more than 100 million years ago. Bedbugs, some of the most unwanted human bed mates, have been parasitic companions with other species aside from humans for more than 100 million years, walking the Earth at the same time as dinosaurs. Work by an international team of scientists, including the University of Sheffield, compared the DNA of dozens of bedbug species in order to understand the evolutionary relationships within the group as well as their relationship with humans. The team discovered that bedbugs are older than bats, a mammal that people had previously believed to be their first host 50 to 60 million years ago. Bedbugs in fact evolved around 50 million years earlier. Bedbugs rank high among the list of most unwanted human bedfellows, but until now, little was known about when they first originated. Experts have now discovered that the evolutionary history of bedbugs is far more complex than previously thought, and the critters were actually in existence during the time of dinosaurs. More research is needed to find out what their host was at that time, although current understanding suggests it's unlikely they fed on the blood of dinosaurs. This is because bedbugs and all their relatives feed on animals that have a home, such as a bird's nest, an owl's burrow, a bat's roost or a human's bed, a mode of life that dinosaurs don't seem to have adopted. The team spent 15 years collecting samples from wild sites and museums around the world, dodging bats and buffaloes in African caves infected with Ebola and climbing cliffs to collect from bird nests in Southeast Asia. Professor Mike Shiva-Jyothi from the University of Sheffield's Department of Animal and Plant Sciences, who was part of the team, said, "To think that the pests that live in our beds today evolved more than 100 million years ago and were walking the Earth side by side with dinosaurs, was a revelation. It shows that the evolutionary history of bedbugs is far more complex than we previously thought." Dr. Stefan Roth from the University Museum Bergen in Norway, who led the study, added, "The first big surprise we found was that bedbugs are much older than bats, which everyone assumed to be their first host. It was also unexpected to see that evolutionary older bedbugs were already specialized on a single host type, even though we don't know what the host was at the time when T-Rex walked the Earth." The study also reveals that a new species of bedbug conquers humans about every 500,000 years, moreover that when bedbugs changed hosts, they didn't always become specialized on that new host and maintained the ability to jump back to their original host. This demonstrates that while some bedbugs become specialized, some remain generalists, jumping from host to host. Professor Klaus Reinhardt, a bedbug researcher from Dresden University in Germany, who co-led the study, said, "These species are the ones we can reasonably expect to be the next ones drinking our blood, and it may not even take 500,000 years, given that many more humans, livestock and pets that live on Earth now provide lots more opportunities." The team also found that the two major bedbug pests of humans, the common and the tropical bedbug, are much older than humans. This contrasts with other evidence that the evolution of ancient humans caused the split of other human parasites into new species. Professor Mike Shiva Jothi from the University of Sheffield added, "These findings will help us better understand how bedbugs evolved the traits that make them effective pests, that will also help us find new ways of controlling them." The researchers hope the findings will help create an evolutionary history of an important group of insects, allowing us to understand how other insects become carriers of disease, how they evolve to use different hosts and how they develop novel traits. The aim is to help control insects effectively and prevent the transmission of insect-vectored disease. The research has been published in Current Biology.